Hi, welcome all the students, undergraduate students of MR Medical College, second year. So this is a pathology class. Here, I'm Dr. Rajesh Patil. So I'll be dealing with the topic HLA system and MHC molecules. So we'll start this particular topic for today's class. So this being the online class. So human leukocyte antigen system and major histocompatibility molecules. So why are these important? Basically, see nowadays a lot of organ failure is there, all the bad habits, all the carcinogens, the cancer incidence as rampantly is very rampant. So all these factors is making more chances for a transplant of any organ, be it heart, be it kidney, be it liver, whatever. So when comes the question of transplantation, then these molecules, the knowledge about these system is very, very important. This is clinical aspect. And apart from that, the exam point of view, the study point of view, the for you all being the students, so this is an important questions that can be asked in your UHS university exams. So definition of human leukocyte antigens. So everyone has several antigens located on the surface of his leukocytes, the WBCs. So one particular group of these antigens is called the HLA, that is human leukocyte antigens. The HLA is responsible for stimulating the immune response to recognize the tissue as self versus non-self, whether it is a self your own or non-self form. So this is a very important point. Is controlled by a set of genes located next to each other on chromosome 6 called the major histocompatibility complex. So HLA and genes that encode them are subdivided into three types again. Okay, the class 1, class 2 and class 3 on the basis of their molecular structure. Class 1 and 2 are structurally similar. Cell surface glycoproteins that are involved in antigen presentation for T cells. So antigen presentation and recognition go hand in hand. So first to be involved in any pathogenetic process, these antigen has to be processed and presented to T cells. That is important. Class 3 comprises an assortment of proteins including several components of the complement system. See this basic chromosome. There's a short arm, long arm, the centromere. So a small fragment from this chromosome 6, it's been displayed there. The fragmentation, the class 1, class 3, class 2. So class 1 has BCA, class 2 BFC2C4 representation, and class 2 DP, D2, DO, DQ, DR. These are the representation of different segments in a particular nucleotide band of chromosome 6. So these glycoproteins that are involved in antigen presentation to T cells. So class 3 comprises an assortment of proteins including several components of the complement system. So this is again important diagram that you have to draw if the question is asked in your university exams. This one, the class 2 DP-DQ-DR fragment segment of that particular fragment of the chromosome 6. The class 3 complement TNF, class 1 BCA. So these are the components of basically the different major histocompatibility antigens, the class 1, class 2, class 3. So basically we came to know about these particular, how these particular major histocompatibility antigens are being classified. So coming to the major histocompatibility, what is the definition of this? The major histocompatibility complex is a large locus on the vertebrate DNA containing a set of closely linked polymorphic genes that code for cell surface proteins essential for the adaptive immune system. These cell surface proteins are called MHC molecules. So genes of the major histocompatibility locus. So again being divided into tabular forms. The gene products, as we already saw about these particular products. The class 1 HLA-A, HLA-B, HLA-C, class 2 HLA-D, class 3 complement C2, C4b, cytokine genes, these are TNF, tumor necrosis factor, alpha, beta. So tissue location, all nucleated cells. So class 1 is expressed on all the nucleated cells of your body, that is important. Class 2 expressed on B lymphocytes, monocytes, macrophages, dendritic cells. So these dendritic cells very rich in the skin. That is nothing but the presentation cell, antigen presenting cell. Activated T cells, activated endothelial cells, the skin Langerhans cells. And class 3 are expressed on the plasma proteins. So these class 3, that is complement C2, C4 and B. So these are contributed by the plasma proteins and even the cytokine genes. Functions, class 1 here, all know where it's expressed on all the nucleated cells. Identification and destruction of abnormal or infected cells by cytotoxic T cells. Identification of foreign antigen by helper T cells. Defense against extracellular pathogens. Cell growth and differentiation. Genetic organization of the MHC, major histocompatibility 1 molecules. The classical HLA spans approximately 3.6 MB and is subdivided into 3 regions. Okay, class 1, class 2, class 3. Each region contains numerous gene loci including expressed genes, transcripts, pseudogenes. There are 3 genes within class 1 region designated the HLA-A, B and C loci. And similarly, there are 3 class 2 genes known as HLA-DP, DQ, DR loci. These were all represented in that particular table in the last slide. So each of these genes exists in multiple different allelic forms and hence HLA complex is the most polymorphic genetic system known. So coming to basically the structure of HLA. Class 1 and class 2 molecules of HLA are membrane bound heterodimers. Okay. Class 1 molecules consist of a heavy peptide chain of 43 kDa called a alpha non-covalently linked to a smaller 11 kDa peptide called beta 2 microglobulin. The largest part of heavy chain is organized into 3 globular domains alpha 1, alpha 2, alpha 3 which protrude from the cell surface. So this is again the representation of the cell membrane, that cytoplasm, how it is protruding out of this particular cell surface. So here microglobulin expression, the extracellular region, so all this. So this diagram is an important one, and how this molecule is being made with this particular smaller CHO molecule, NH2, the hydrogen, the nitrogen component, COOH. So all these molecules. So this is protruding from outside the cell membrane. So that is the class 1. The largest part of heavy chain is organized into 3 globular domains. Okay. So X-ray diffraction analysis of crystallized class 1 protein has revealed its 3-dimensional structure. So most of the molecules has a beta pleated sheet structure, but alpha 1 and alpha 2 domains contain a alpha helix. So this is again the class 1 MHC molecule diagram representation, the alpha 1, alpha 2, alpha 3, beta 2 microglobulin. So these are the expressions here. Class 2. The class 2 proteins HLA, DP, DQ, DR are also composed of 2 non-covalently associated polypeptides and both chains are encoded by polymorphic genes located within HLA complex. So this is again the representation of this particular thing, how it is being protruded out here there. Each class 2 protein consists of an alpha chain, beta chain, which are both anchored in the cell membranes. X-ray crystallography has shown that the alpha 2 and beta 2 domains, the ones nearest to cell membrane, assume characteristic immunoglobulin fold, while alpha 1 and beta 2 domains form a groove bounded by two alpha helix and beta pleated sheet floor. So this is again the diagrammatic representation. So differences between MHC class 1 and class 2. So important one, it can be asked as a even the 3 marks question. So apart from the main question, So this is how this is the cell surface, the cytoplasm, and how these molecules are protruding out of the cell wall. So here class 1 molecule and class 2 molecule. So peptide binding cleft is there here on the representation, membrane distal domains, membrane proximal domains, and here the alpha 1, the alpha 2, and structurally there is some variation there. So the cleft, complete cleft here in class 2 and partial cleft in class 1, but both are bound here. The binding of this class 1 with a single string there, but double string in class these particular two molecules. Okay. So immunobiology of the HLA system. Coming to the immunobiology. HLA molecules are membrane-bound glycoproteins that bind processed antigenic peptides and present them to T cells. Gene inheritance follows Mendelian principles with N-block HLA A, B, C, DR, DQ haplotype transmission from each parental chromosome, although recombination within the HLA system can occur. All of the inherited antigens are codominantly expressed. HLA class 1, A, B and C antigens are expressed on almost all nucleated cells and platelets and present intracellularly processed peptides of viral or self origin to CD8 plus cytotoxic T cells. T cell recognition of a non-self or with a self HLA class 1 molecule can result in a cytotoxic T cell response against the target cell. HLA class 2, DR, DQ and DP molecules are expressed on a more restricted range of the cells, including B cells, activated T cells and the monocyte macrophage lineage. So that table that I described on the last slides are really important. Which cell express which molecule? The nucleated cells or the expression on all the cells. Class 2 molecules present peptides mostly of exogenous origin to CD4 helper T cells. So here again a table, class 1, class 2. Such tables are very much essential for you to learn, and when you represent your answer on the answer sheets in the exam, such tables fetch you good marks. So here again this particular table, all nucleated cells, antigen presenting cells, the expression of these class 1. So then peptides, the CD8 plus T cells, the expression of class 1 here, the class 2 on CD4, the antigen presenting cells. The amino acids, number of amino acids involved in each 8 to 10, 15 to 18. So endogenous, intracellular, exogenous, alpha 1, alpha 2 groove, alpha 1 and beta 1 groove, CD4, CD8 binding site, alpha 2 binds to CD8 molecule, whereas beta 2 binds to CD4 molecule. Antigen presentation pathways. The cytosolic pathway, this one is the endocytic pathway. So the histocompatibility between individuals is based on the extent of matching of inherited HLA antigens and the degree of immunologic reactivity to these antigens. See, that's the reason, say for example, I still remember an event wherein a doctor had met with an accident, and lot of there was no blood supply to one of his kidney, which was noticed very later post traumatic time. It consumed lot of time, so lot of necrosis and gangrene had happened in the kidney, and the kidney has to be removed. So and another kidney had severe has been damaged severely. So what happened in him was both the kidneys had to be removed, and who was the immediate person who can donate to him? HLA matching has to be done. So his own elder brother donated it. Both are from the big VIP families. See, however money you have, however VIP you are, but in that particular time, that particular day, you should, when you have to have a kidney, HLA matched person should be available and ready to donate a kidney to you, or whatever. Only the patient can be revived or will survive, or else will succumb if no one gives the organ, or if a person is ready to give the organ and HLA doesn't match, what's the use? So immediate family member, the first degree relative, most of the times matches with this or has the common HLA matching system. That's the reason immediate donors are the first degree relatives. So cross matching has to be done. HLA matching and histocompatibility testing have an enormous practical influence on contemporary organ transplantation practices in clinical medicine. Histocompatibility testing. The practice of testing for histocompatibility between an organ donor and the selected recipient is based on the presumption that tissue compatibility will promote graft acceptance and avoid immune rejection. The foundation for this assumption is the evidence that identical twins can accept and retain grafts from one another indefinitely, whereas grafts from all others are ultimately rejected in the absence of immunosuppressive therapy. So immunosuppression, usually is provided in transplanted organ patients, so that rejection shouldn't be there, or they are put on high doses of steroids. So identical twins, if you are an identical twin brother or anything, you are a lucky person, because if your organ fails, then you have another twin who can readily donate you some organ from his body to you, because lot of HLA matching is there. So this is another diagram. Principle used in histocompatibility testing. What is this principle? HLA antigens are identified using sera with known anti-HLA antibodies and DNA-based molecular typing. Recipient serum is screened for the presence of HLA antibodies using immunoassays. Compatibility is directly determined by cross-matching of donor cells and recipient sera. So this is a beautiful flow chart being displayed here. Tissue typing has 4 connected functions relating to the MHC and transplantation. It defines the human leukocyte antigen type for the matching of donor and recipient organs, that is MHC matching. It defines which antigens a recipient cannot be given because of previous responses induced by pregnancies, transfusions, transplants, etc., and hence indicates which mismatches may safely be given windows. So this particular thing, so some have a ready acceptance, but some have always the rejection due to mismatch of this particular HLA. It checks at the time of transplantation that no humoral response is present against a donor MHC antigen cross-matching, and it defines when and to which MHC antigen a person responds to after transplant induced responses. This particular test is frequently used to confirm apparent HLA identity in the living related transplant situation. When the HLA identical recipient and donor are unrelated, as in voluntary bone marrow donation, MLC testing is extremely important in revealing hidden class 2 incompatibilities that could affect recipient intolerance to the graft and promote graft versus host disease. So these are very important one here. So the human leukocyte antigen. Tissue typing method classifications. Serological techniques, complement dependent cytotoxicity, panel reactive antibody, and then typing by cellular response, mixed lymphocyte reaction. 3. Typing at the DNA level, fragment based methods, that is restriction, fragment length, polymorphism. PCR based methods, probe hybridization methods, PCR using sequence specific oligonucleotide probing, direct amplicon methods. So all these PCR systems. PCR using a sequence specific primers. Second classification, determining HLA type antigen. Now how do you do that? Detecting variation in expressed HLA molecules, that is the serological and cellular typing. Mixed lymphocyte reaction, mixed lymphocyte culture, complement dependent cytotoxicity test, homozygous typing cells, and primed lymphocyte typing, that is PLT. So some abbreviations are used here. These are not the standard ones but the convenient ones just on the PPT sake. Cell mediated lympholysis assay. So detecting variation at DNA sequence level, that is DNA typing. Restriction fragment length polymorphism, RFLP. So these are not the standard abbreviations or the standard short forms as such, but conveniently used here. PCR with sequence specific primers, PCR with sequence specific oligonucleotide probes, PCR with sequence based typing, heteroduplex formation, variable number tandem repeats. So these are the nucleotide base pair repeats, variable number of tandem repeats. That's also typing is required. Then 2. Antibody identification. HLA antibody screening by complement dependent cytotoxicity. HLA antibody screening by ELISA. HLA antibody screening by flow cytometry. Then the class 3 one, patient donor cross matching. Cross matching by complement dependent cytotoxicity, cross matching by flow cytometry, cross matching by solid phase methods. The antibody identification. Exposure to HLA antigens can occur as a consequence of blood transfusions, prior organ grafts, or pregnancy. The resultant formation of specific antibodies to those antigens is termed sensitization. Re-exposure to the previously immunizing antigens on a new allograft can produce rapid humoral and cellular immune responses, leading to hyperacute or accelerated rejection. Detection of such preformed specific antibodies is of paramount importance in evaluating the state of initial histocompatibility between recipient and donor, especially in the face of known HLA antigen mismatches. The simplest and fastest methods for histocompatibility testing are serologic, that is, they utilize blood serum that contains antibodies to HLA antigens. This is an important flow chart here. The sera containing HLA antibodies plus mixed with lymphocytes. Then the antibodies bind to their specific target antigens on the cell surface. The antigen antibody complex is formed on the cell surface. These particular expression, the molecules are expressed. These are protruding out of the cell surface from the cytoplasm, from the cell membrane, and protrude out of the cell membrane. The expressions, these particular antigens, and They are the histocompatibility antigens that bind and create this antigen antibody complexes on the cell surface. So there is that leads to complement activation leading to cell lysis. Thus, cell death is an indicator of the shared specificity of antigen and antibody and is a positive test result. Application of HLA typing, organ and tissue transplantation. So this is very important one, the first step, and then comes the forensic medicine one. Determining HLA haplotypes can also be crucial for the gathering of forensic evidence and for studies of anthropology. So this is the clinical application aspect. Diagnosing some diseases like autoimmune diseases and viral diseases. In autoimmunity, many HLA combinations are potentially indicative of autoimmune disorders. Example, so these are the classic examples here. The HLA-B27, that is ankylosing spondylitis, reactive arthritis, Reiter's syndrome. Then the allele, the HLA allele DR2, that is associated disease, multiple sclerosis, good pasture syndrome. So this can be asked as a short notes also, 3 marks question also, which are the HLA alleles and related diseases. So even in Viva. Then DR3 allele is related with the myasthenia gravis. The superstar of Bollywood, Amitabh Bachchan, suffers from this. As you all know, myasthenia gravis, but still he's so hale and healthy, even though with myasthenia gravis. So life is all about how you deal with different things, or the how you react to these particular diseases that you have with positivity. The positive energy, the positive thought really doesn't hamper your lifestyle or doesn't impair your health, and you just live hale and healthy. The classic example is that of Amitabh Bachchan. DR3, DR4, this is again the allele which is involved with the diabetes mellitus. DR4, rheumatoid arthritis. A3B14, this is again the allele associated with the hereditary hemochromatosis. So we know all these HLA alleles related with different diseases. Susceptibility to viral infections. How susceptible a person is. Nowadays everywhere corona, COVID-19, as you all know. So this is a very dreadful pandemic that we are come across. Last 1.5 years, all colleges, schools are hampered. So this particular virus, is creating a ruckus, is creating catastrophes in families, in society, in country, in the world, everywhere. So how susceptible you are to this particular, I know a few families wherein wife succumbed to this particular COVID virus, and husband sleeping with the wife is hale and healthy, and it is the husband who is a doctor and wife is a housewife, and the husband gets exposed to COVID patients on a daily basis. He is a famous radiologist who does CT scan for these COVID patients, and he is hale and healthy and is negative, COVID negative, but when the same wife who shares bed with him every night turned positive and the husband is negative. Now what could be the reason? In this scenario, in this family, the reason could be attributed most of the times to his health, good health, and his susceptibility threshold. So only thing we can say is that he has got very good high threshold and is not susceptible to such infection. So that's an example of here susceptibility. There is a link between certain HLA antigens and susceptibility to some viral infections. HIV, hepatitis B, hepatitis C, all these infections also, it all depends upon the susceptibility. The rubella. Paternal testing, another important applied aspect. HLA typing can be used alongside other tests for paternity testing. So important medical legal aspect here, the HLA typing one. The infertility, recurrent pregnancy loss in such cases. due to recurrent pregnancy loss can be attributed to immune factors, one of which is presence of certain common HLA antigens between the parents. Phylogenetic studies. Some HLA haplotypes have distinctive geographical distribution and are found only in some population. These haplotypic types can be used to trace human migration. Mechanism. The pathophysiology of graft versus host disease includes three phases. The afferent phase, activation of APC antigen presenting cells. The efferent phase, activation, proliferation, differentiation, and migration of effector cells. The effector phase, target tissue destruction. Activation of APC occurs in the first stage of graft versus host disease prior to hematopoietic stem cell transplantation, radiation, or chemotherapy. Results in damage and activation of host tissues, especially intestinal mucosa. This allows the microbial products to enter and stimulate pro-inflammatory cytokines such as interleukin 1, tumor necrosis factor alpha. These pro-inflammatory cytokines increase the expression of MHC and adhesion molecules on APCs, thereby increasing the ability of APC to present antigen. The second phase is characterized by the activation of effector cells. Activation of donor T cells further enhances the expression of MHC and adhesion molecules. Chemokines and expansion of CD8 plus and CD4 plus T cells and guest B cells. In the final phase, these effector cells migrate to target organs and mediate tissue damage, resulting in multi-organ failure. Now, coming to another important 5 marks question and clinically important one, transplant rejection. So, according to genetic relationship between donor and the recipient, transplantation of tissues is classified into 4 groups. Autografts. Now, these are grafts in which the donor and recipient is the same individual. Isografts are grafts between the donor and recipient of the same genotype. So it is the same this thing race. Allografts are those in which the donor is of the same species but of a different genotype. So the genotype is different here. Xenografts, less highlighted points usually and less studied, I think so, are those in which the donor is of a different species from that of the recipient. So a lot of research work are going on here. Just imagine, what if a dog's liver can be put in a human liver without rejection? Oh my god, most of the dogs might have been killed and most of the humans be saved. But that's pity on dogs, on animals if that is successful, and good news for humans. Really can't comment much on positive thing or negative thing on that particular point, because you're killing some animal to save a human race there again. So coming to cell mediated immune reactions, these are mainly responsible for graft rejection and are mediated by T cells. The lymphocytes of the recipient, on coming in contact with HLA antigens of the donor, are sensitized in case of incompatibility. Sensitized T cells in the form of cytotoxic T cells, as well as by hypersensitivity reactions initiated by T helper cells, that is CD4 plus, attack the graft and destroy it. Humoral immune reactions. In addition to the cell mediated immune reactions, a role for humoral antibody in certain rejection reactions has been suggested. These include preformed circulating antibodies due to presensitization of the recipient before transplantation, that's a classic example, by blood transfusions and previous pregnancies. or in non-sensitized individuals by complement dependent cytotoxicity, antibody dependent cell mediated cytotoxicity, that is ADCC, and antigen antibody complexes. So this is again the diagrammatic representation here, the direct pathway, the indirect pathway of how class 1 MHC molecule, with the binding of this particular surface receptors, the antigen presenting cell in the graft, how this class 2 MHC molecule cell surface are being expressed here, and how these particular CD4 T cells are being stimulated here. CD4 helper T cells, renal blood vessels, CD4, CD8. So the activated macrophages here, the CD4 binding comes, and then affects the renal tubule. So all this is the schematic representation. See this renal blood vessels containing the antigen antibody complexes. So this is, such diagrams should be used in your answer sheets in the exams if the question asked. Types of kidney graft rejection reactions. Based on the underlying mechanism and type period, rejection reactions are classified into three types: hyperacute, acute, and chronic. Hyperacute rejection Hyperacute rejection appears within minutes to hours of placing the transplant and destroys it. It is mediated by preformed humoral antibody against donor antigens. Cross-matching of the donor's lymphocytes with those of the recipient before transplantation has diminished the frequency of hyperacute rejection. So this is again the hyperacute rejection here. So cross-matching of the donor lymphocytes with those of the recipient before transplantation has diminished the frequency here. So what happens here? Grossly hyperacute rejection is recognized by the surgeon soon after the vascular anastomosis of the graft and is performed to the recipient's vessel. So after the vascular anastomosis of the graft is performed. So this is what happens here. Basically, the organ becomes swollen, edematous, hemorrhagic, purple and cyanotic, rather gaining pink color. Histologically, the characteristics of arthrous reactions are present. There are numerous neutrophils around dilated and obstructed capillaries which are blocked by fibrin and platelet thrombi. Small segments of blood vessel wall may become necrotic and there is necrosis of much of the transplanted organ. So necrotic organs are very dangerous. So it can cause to septicemia and gulp the life of the patient. So very tricky situation for the handling physician or a surgeon there. So acute rejection. This usually becomes evident within a few days to a few months of transplantation. Acute graft rejection may be mediated by cellular or humoral mechanisms. Acute cellular rejection is more common than acute humoral rejection. Microscopically, what you see? The features of the two forms are as under. The acute cellular rejection is characterized by extensive infiltration in the interstitium of the transplant by lymphocytes, mainly T cells, a few plasma cells, monocytes, and a few polymorphisms. So these few polymorphs are nothing but the neutrophils one. There is damage to the blood vessels and these are foci of necrosis in the transplanted tissues. Acute humoral rejection appears due to poor response to immune suppressive therapy. So, rejection. So whenever there is a transplanted organ in the body, whatever nowadays, the heart, the living heart, and some patients even they have experiment with the double heart system. One not so active heart, and the another one very active transplanted heart. So that's also there. So that's a live that was performed recently. Organ was taken from Bangalore, shifted to Chennai operation theatre, and the surgery of placing another second heart was done in Chennai, the living heart transplant. So these post-transplantation patients are given with a high doses of immunosuppressive agents so that rejection doesn't happen. Chronic rejection. So here what happens, chronic rejection may follow repeated attacks of acute rejection or may develop slowly over a period of months to a year or so. The underlying mechanism of chronic rejection may be immunologic or ischemic. Patients with chronic rejection of renal transplant show progressive deterioration in renal function as seen by rising serum creatinine levels. Microscopically, in chronic rejection of transplanted kidney, the changes are intimal fibrosis, interstitial fibrosis, and tubular atrophy. Renal allografts may develop glomerulonephritis by transmission from the host or rarely may develop de novo glomerulonephritis. So this concludes this session that we came across this histocompatibility antigens, the HLA system, that is human leukocyte antigen and major histocompatibility molecules. So this is an important point. Just go back home and revise this. This can be asked as a big main question or even as a short notes, 3 marks. So it's an ideal slot for a 5 marks question, but 3 marks also form a chunk of this story. Thank you. Dr. Rajesh Patil signs off from this particular session. The session ends. Thank you. Have a nice day.